SARS-CoV-2, since the advent of the coronavirus in 2019, we've seen that it has caused a lot of deaths. And as it stands now, about 6 million people have died, and more than 470 million people actually infected. But if you look at the pandemic when it started earlier, you realize that Europe as kind of the epicenter where you have a lot of cases. And then, but if you look at it generally, you'll see that the pandemic has evolved differently around the globe. For example, in Africa, we still saw quite number of deaths, like the number of deaths and cases is quite low compared to what we see in America and Europe. And there are a lot of speculation and theories regarding the reason why this is happening. But what is worth mentioning is that this might be underestimated, given that many countries don't have the ability to sequence or do sequencing in Africa. What is also interesting is that during the pandemic, we've seen variants coming in, introduction of variants due to mutations, which are seemingly responsible for what we see, increase in disease transmission, disease severity, as well as even evasion. Classical example is the B.1.17, which is also called the alpha variant. It's been reported that up to 90% more transmissible than the existing variants. And this also been seen with the Delta variant, also called before the Indian, but now it's called the Delta variant, which is given the B.1617 lineage nomenclature. Here we've seen that 50% more transmissible than the alpha variant. In fact, the advent of this variant in Africa has actually changed the dynamics, because we've seen so many countries reporting a lot of deaths and a lot of infection during this wave. So if you don't know the Gambia, I just want to give a brief introduction of where the Gambia is. It's located in the coastal side of West Africa, surrounded by Senegal on all three sides. And then it's a very small country with 2.5 million people, about 17.5 years median age, very young population, and half of the population, more than half of the population, live in the coastal sides. And here, if you can see my mouse, is where you have the MRC unit, the Gambia at the coastal side, and this is where you have majority of people living. It's a subtropical climate where you have more of dry season and few months of rain and wet. I also want to mention that it's a tourist country where you have very nice sandy beaches, and that's the reason why when the borders were open, we've seen a lot of cases in the Gambia, SARS-CoV-2. So in the context of what we're going to talk about, it's important to understand how the system works. The health system is in the Gambia. The Gambia government actually is responsible for the welfare of the health system. And we have about 4 referral hospitals scattered all over the country. And then majority of the hospital and the health centers are within, and we also have about more than 30 non-governmental organizations, which MRC is part of, operating in the country, and small other clinics, private clinics in the Gambia. So the Genomics platform is actually, we're very lucky. I started working here in 2017 when I just finished my MSc in bioinformatics. I joined Dr. Abdoulie, who is the head of the lab. And here we do a lot of sequencing, and we collaborate with scientists and inside the unit and outside. And we sequence a lot of different type of samples, ranging from bacteria, viruses, as well as now we're thinking of doing human genome sequencing, because we have the capacity to do so. And I'm very happy to mention that since the the epidemic have started in the Gambia, we've been sequencing SARS-CoV-2 in the unit, and more than 1,000 genomes have been deposited in GISAID for public use. Our workflow is not very different from what is available in many labs. It's very simple. Sample reception and extraction is actually done at the National Public Health Lab. We collaborate with the government on that. And then inside the unit here, we also have a molecular diagnostic platform that does the extraction of RNA, which are sent to the genomics platform for library preparation and sequencing. Sequencing is actually done more on the nanopore platform for turnaround time, because we know the nanopore is really good when it comes to having the sequences very quickly. And that is really good for reporting the variants that are available. So we also have the HPC, which is a high power computing infrastructure in the unit, which we use to do most of our analysis. So here, the figure here is just to show that we do some quality checks at any point in time. We, the molecular diagnostic platform, or the people that do the purification, change some of their the target genes. We try to check to make sure the quality, what we see with our genomes is actually correlating with what they're giving us as CT, because CT values will definitely vary from lab to lab. So as I alluded previously, we've been using the ARTIC protocols to do most of our sequencing. This is based on 93 primer pairs initially, and these have been updated as the pandemic evolves. And we've also realized that at some point, because of mutations, we have some drop out in some regions, and that's the reason we keep updating the primers that we're using. Here you see the midnight primers and the VAR script, which we've used currently to make sure we don't miss any variant or lineage that comes into the country. So I just now want to mention some of the work that we've been doing and how it impacted the way we see SARS-CoV-2 in the Gambia. So with the first SARS-CoV-2 that was identified or reported in the Gambia was in March 2020. And when this happened, we also see a few cases that came in. So what we did, we quickly sequenced these samples, and then we used phylogenetic analysis to describe what we saw. What we saw was that all the cases were actually imported from Europe, and majority of them came from UK and France. So when this was published, or when this was reported to the government, a state of emergency was declared, and all businesses and movement of people were restricted for more than 5 months. But what is interesting is that during the lockdown, we've realized that from July, we've started seeing increase in number of cases. And this was really surprising, and I'm part of my PhD because I'm doing it currently. When I looked at the data, what we saw is that a majority of the samples that are coming into the country might be coming through Senegal, because we are very close to Senegal, and the land border was actually very porous. And when we look at the genomes, we actually saw that this hypothesis was justified from the genomic analysis, because we've seen high prevalence of this particular lineage we call the B.1416. And this lineage is high prevalent in our setting in Senegal and Gambia. Only 2 countries in Africa, which is Morocco and Burkina Faso, reported only 2 sequences from each of these countries. So we kind of thought that this probably might be the reason why we were having surges of cases, even though we were on state of emergency. So from the beginning, we keep doing analysis as we got samples coming in, and these were some of the samples that we sequenced in October from March to October 2020. And what we see here is that we had few varieties of lineages that were circulating in the country, and majority of them are actually coming from Europe. It wasn't that different from what we saw in Europe. So it basically tells us that the genomes are actually coming more from Europe than any other country. So this particular, as we go into the pandemic, we also do more analysis, and we always try to make sure part of our role is to ensure that the variants are actually detected at the right time and reported to the government. So in May 2021, we saw that the alpha variant is taking over. About 70% of all the positive samples that we were sequencing actually coming were the alpha variant type. And so this was only possible because we had the sequencing done real time, making sure the samples reach the lab at a very early stage, and then sequencing was also done and reported to the government and deposited in GIS as well. So this is another classical example. In April, late April, we saw 2 people that came from India, and we quickly sequenced their samples. Luckily for us, these people came in, and only after 2 days we were detected. They were not positive on arrival, but after 2 days they got symptoms, and samples were taken, sent to the lab for sequencing, and that's when we detected the Delta variant. So the important part here is that we were able to go and get all the contacts and the cases and sequence all their samples to ensure that this doesn't transmit quickly to the community. But as we know, the Delta variant was really very transmissible. And of course, at some point, it overtook the Alpha variant during its wave. We worked on with a lot of people during the COVID. I didn't mention about our travels, but during the COVID, myself and my boss and the team, we traveled to a lot of African countries to ensure that we establish sequencing facilities within the region, because it's important that if we are sequencing and we know what variants are in the country, but we don't know what is happening in other parts of Africa, it doesn't really mean anything. So we were able to do that in support of MRC and Fitch. We got some grant, and then we used that money to invest on labs that are within the unit. And some of these publications are actually done because we had the funding and we have the sequencing facility in MRC. So I just want to wrap up that this work was not possible without the dynamic team we have. We have very talented individuals in the lab that does all the sequencing. I do most of the analysis and of the using bioinformatics, but majority of the wet lab is done by these gentlemen and women that you see on this page. And Abdul Karim is the head of the genomics team in the Gambia. I thank everyone that have contributed towards this sequencing in the Gambia.
Hi, I'm excited and very honored to be part of this, the first Human Cell Atlas Africa Symposium. I am a clinician in pediatrics and infectious diseases. I'm at the University of Glasgow, but based most of the time in Malawi, where I work at Queen Elizabeth Central Hospital, Malawi Liverpool Wellcome Programme, and KUHES. We have a couple of ongoing single cell studies, but today I'm going to present you some of our COVID data because I think this study was the most current. We've had 4 waves in Malawi based on different variants: the ancestral variant, beta variant, Delta variant, and Omicron. And this study was conducted between, we collected samples between October 2020 and June 2021, so during the beta and Delta variant waves. At the time we started, back in October 2020, there was many questions about COVID in any setting, but we really had very little understanding of what COVID-19 was in an African population, like the patients we were seeing in the hospital in Malawi. And we therefore thought it was really important, as data were emerging and more understanding was emerging in Northern Hemisphere settings, to do a systematic assessment of the pathology in a Malawian setting to compare with other cohorts. And really, COVID is a disease that's significantly or mostly dominated by disease in the lung, and the lung is only really accessible post-mortem. So we did this using an autopsy study, firstly to do systematic assessment of histopathology, and then to dive down more deeply into understanding the cell states and cell compositions of the lung, and then also of lung and nasal cells in the same patients, which are cells that would be accessible from patients during life, because it's so challenging to get these data from post-mortem studies. Important to understand what these accessible samples during life tell us about what's going on in the definitive organ, the lung. And then finally, we aim to conduct an analysis to understand the key molecular pathways and cellular pathways governing disease in the lung in our setting. And this is important because now that more and more data on the efficacy of immunomodulatory treatments in northern settings is available, it's really important to know whether similar cellular and molecular processes are at play in our population, or whether there's really distinct immune processes involved. Unfortunately, I can present you data from the first three. This one is very much ongoing, really just started, so that will have to be for the next talk. Fortunately, at the time of COVID, we were already conducting an autopsy study to understand causes of death in children. We were using minimally invasive autopsy, which uses large bored needles to take biopsies from all vital organs. This technique is really useful because it's much quicker than a full autopsy, much more acceptable to families because it leaves only pinpoint marks. And from an infection point of view, there's much less infection risk because you're not opening any body cavities. There were still substantial challenges to conducting such a study during the kind of COVID. We had to have extensive talks with the ministry and with senior people at the hospital and conduct sensitization in 150 staff on the acute medical wards, from cleaners through to clinicians. And then we did refurbishment of an area of the mortuaries to be able to conduct COVID-specific autopsies, and did training of our staff to ensure safety during autopsies. This was really a very challenging undertaking indeed, and involved an extraordinary team. I don't have time to credit the fantastic efforts of all of these people. We had a diverse range of skills, from social scientists, clinicians on the ward, PIs who are clinicians in other settings with expertise in autopsy studies, and then a number of people who did the work in the laboratory. We recruited 16 fatal cases that we did autopsies on. We defined these antemortem when we recruited them on the basis of clinical information, distinguishing COVID pneumonia, who had SARS-CoV-2 positive tests and clinical or radiological evidence of pneumonia, pneumonia cases who are not SARS-CoV-2 positive, and then patients who had no evidence of pneumonia clinically or radiologically antemortem. We then, including the detailed histopathological assessment, had a cause of death panel consisting of a number of experts to definitively assign a cause of death, including all the information. And what you can really see here, the first take home is that the clinical team on the wards in our hospital are really good at picking up COVID-19 antemortem. So when they thought it was COVID and treated it as such antemortem, we thought it was COVID in the panel, all of the additional information that we had. And there were no cases that we, with the additional pathological, detailed histopathological information, thought had COVID pneumonia who were not picked up to have that antemortem. Highlighting some features of our population. Generally, the CT values were pretty low. The patients were younger on average in the other cohorts and had a much higher rate of HIV positivity. And these were consistent with what we've seen more broadly in our patient population at the hospital, at Queen Elizabeth Central Hospital. The COVID cases did have traditional comorbidities associated with COVID: obesity, hypertension, and diabetes in all cases, and sometimes all three. Some of the cases were, the first 9 cases were during the beta variant, and the remaining cases were during the Delta variant. So we did a detailed assessment of histopathology in multiple organs, in particular in the lung, which had by far the most interesting findings. These were assessed by Steve, Kamiza, pathologist in Malawi, and then 2 independent pathologists, Cheryl Lampos and Vanessa in the UK. We picked up, the most marked feature was changes to the alveoli, hyaline membranes that you can see here, diffuse alveolar damage, and emphysema, edema, and hyperplasia of type 2 pneumocytes. This heat map shows the COVID cases at the top, pneumonia cases, and then control cases, each patient being a row, and the column being the different features scored from 0 to 3 by the pathologists, and then displayed as a heat map. So then drilling down more deeply to the cellular level, we did single-cell RNA sequencing and FACS. Single-cell sequencing was done by Charles and James in my lab, analysis by Thomas Otto in Olympia in Glasgow in collaboration with Benjamin Kumwenda at Kuhes. And then Kondwani Jambo's team led multi-parameter flow cytometry, James and Leonard, PhD students of his in the lab. The data I'm going to show today are from our later six patients. We initially planned to use slow frozen samples to do single cell RNA sequencing, but while this worked well on control cases, it was really very poor in the COVID cases. I think that's very consistent with what others have found. And so we did fresh samples for the last 5 cases, and then we have done single nuclei cell partitioning for the remaining cases, but we're still awaiting sequencing for all of those data. And I'm just going to show you the whole cell data today. Despite the fact that we only have 1 COVID, 2 pneumonia, and 2 non-pneumonia, we do have a good representation of stromal and immune cells, including 3 different neutrophil populations and a number of different macrophage populations. I've only differentiated here pneumonia, including both COVID and non-COVID, from the non-lung disease controls. And you can see that there's much more neutrophils, as you'd expect in the pneumonia and COVID, and some differences in the macrophage populations. Moving to the nasal cells, this is from 3 cases from each group. You can see also a good representation of stromal cells and then immune cells, some of the same macrophage populations, and then T cells and B cells. You can see that T cells are almost completely absent in the COVID cases, but we do see a significant number of neutrophils, and all have a representation of the stromal cells. And then finally, in whole blood, from 2 cases per group, you can again see a good representation of different cell types. In whole blood, so we have neutrophils, 4 different neutrophil populations. Unsurprisingly, these were highly enriched in the COVID cases in blood, including in particular immature and highly inflammatory subtypes. And we also saw, I should say, a depletion of T cells in the blood samples. And then moving to our flow cytometry data, which is a little bit further along. This largely mirrors what we saw in the single cell data. But here, with a limited amount of time, I just want to highlight one thing that was really very marked in this data. So these are t-SNE plots with clustering based on 14 different antibody markers to characterize, in this case, myeloid cells. We had a myeloid T cell and a neutrophil panel. You can see that some of the cell types that were present in the lung and the nose, some of the cell clusters were not present in blood. And then this is comparing COVID and non-COVID, and then lung, nose, and blood make up the columns. These 4 different squares show heat maps for 4 different markers. And in particular, I'd like to highlight that CD206, a marker of specific macrophages, particularly present in tissue. You can see, as expected, but importantly, that these are present in the lung and the nose, but completely absent in blood. This was a consistent pattern throughout the data in the different panels. And this is really validated by Luminex data that we did on nasal fluid and plasma, looking at a number of different cytokines and chemokines expressed here, shown here on different rows, and then the patients are the columns. You can see that there's clustering, particularly among COVID cases and some of the pneumonia, with upregulation of inflammatory cytokines and chemokines in the COVID cases, but not the controls in the nasal fluid, particularly those from chemokines and cytokines from neutrophils and myeloid cells. But then there really isn't a convincing clustering by patient group in the plasma, comparing these 2 fatal, very sick groups of patients. And finally, not time to go through this in detail, and we're only at the beginning of analyzing it. In all 16 cases, we've done imaging mass cytometry from 4 to 8 lung regions, regions of interest per patient, based on tissue microarrays and 35 different antibodies. And this will then allow us, in a subset of markers, to map some of the immune interactions and cellular states onto the tissue, and to compare with our single cell data. Just a teaser of data here from one region of interest, looking at some of these different panels because we can't see all 35 markers by eye. This is the pneumonia on the top row and COVID on the bottom. You can see infiltration of different neutrophils in both pneumonia, more in pneumonia than COVID. Myeloid cells here, tissue macrophage markers, in fact, which are more present in the COVID cases. And this here are showing really marked fibrosis, looking at smooth muscle actin, collagen, and fibrinogen. So some indication that these data have biological meaning. We saw a reassuring concordance between antemortem and postmortem data, showing that we aren't missing a huge swathe of COVID on our wards. Our patients were younger than other cohorts, high HIV rates, and a rapidly progressive illness, and had pathological and immunological similarities with other populations, but also differences. And we will drill down into those in much more detail over time with more data. Our data so far suggests the nose is useful for understanding the lung pathology, but the blood is not. And we did see myeloid dysregulation in all compartments. So we think, most importantly, this is a useful platform for investigating other diseases and contributing to the Human Cell Atlas, including my ongoing work on brain infection. And most importantly, I want to highlight, we're very happy to collaborate and share protocols and experiences, including our successes and mistakes from what we've learned. I want to acknowledge this wonderful team, who I don't have the time to credit them all, and also Alex Shalet for providing technical tips and even a reagent for our single nuclei sequencing. Thanks, Alex.
So today I'm just going to give a brief overview of some of part of my PhD research. So just trying to assess the cellular changes during the blood stage of malaria infection using a controlled human malaria infection study. So to begin with, malaria is quite a big problem in sub-Saharan Africa due to the geography of the land, since most areas are tropical and subtropical. So we get varying malaria transmission intensities. So what you do find within this region is that residents in these endemic areas over time develop some form of immunity. So this immunity is slow to develop and requires a number of malaria episodes. And this immunity as well is not sterile, so it doesn't protect you from subsequent infection. You can still get infections. The only upside is you don't get the clinical symptoms, and sometimes some individuals are able to control parasitemia. And this was showed by Cohen in 1961, where he took antibodies from malaria endemic, from adults in a malaria endemic area, and they were given to children undergoing a malaria infection. And what he saw was that there was a decrease in parasitemia as well as the clinical symptoms of malaria. So that showed that antibodies are key in controlling the malaria parasites. But then the process of antibody production in the body is a multi-step process. It requires the coordination of different cells. The key being the dendritic cells, which are the body's main antigen-presenting cells. So they present malarial antigens to T cell, specifically CD4 T cells, causing them to expand, and they provide help to B cells, causing them to class switch and produce antibodies. So the biggest issue is that during a malaria infection, dendritic cells are hampered, so they don't function well, and this interferes with the dendritic cell and T cell interaction. Basically, we get this impairment of antibody production. So what we wanted to do was to understand how this works, and to do that, we wanted to use an in vitro system. So we used the Controlled Human Malaria Infection model that has been set up at the Kemri Wellcome Trust in Kilifi. And so our samples mainly came from the coastal region, 2 areas that are not that far apart, but then they have varying transmission intensities. So this region called the Ngerenya region has a moderate transmission of malaria, while Njunju has a high transmission of malaria. So what we do is we recruit participants. So these participants have a varying levels of antibodies towards the malaria parasite. So what we do once they're recruited, we screen for the antibodies using Schizont extract. After that, we take a blood sample a day before they're inoculated with sporozoites, and then blood samples are taken at the specified days until day 21, which is the endpoint of the CHMI study, and all participants are put on anti-malarial drugs and followed on just to make sure that they don't have any parasites in the blood system. As well as that, there is a sample that is taken twice daily just to try and see how the parasites are growing during the process of the challenge. So using the parasite growth data, so from the PCR, so you get these 2 populations, which you can't see clearly here but we can separate them. And then what you see is that you have one population where after they're challenged with sporozoites, you see that they slowly increase in parasitemia over time. So we took these population, so because they're not able to control the parasite. Once they're injected with these sporozoites, they slowly develop malaria symptoms, as well as being blood positive by PCR and as well by blood film examination, so by microscopy. And then the other population you see is this population of people who are able to control parasitemia below the threshold of the CHMI study. And these participants were able to stay in the study until its endpoint at day 21 days post-challenge without showing any clinical symptoms, but they had detectable parasites by PCR. So once we selected our samples that we're going to use, we had then again to select the time points. So the first time point was the one here, just shown here, sorry. The first time point was at day T-1. So we chose this time point basically because at this time point, everyone is basically the same. No one has any malaria parasites in the bloodstream. The only thing they have is the pre-existing immunity, which varies depending on which region you come from. And then again, the next time point we took was 9 days post-challenge. So at this time point is when the malaria infection is establishing itself in the blood. And then we chose day C+14 because majority of the febrile population were treated at day 15 or day 16. So we chose a time point where we had all participants still enrolled and they were not being taken off due to the levels of parasitemia in the blood. So those are the 3 time points we had, and then these are the samples that we picked. So from that, what we did is we took the samples for single-cell sequencing, and in order to cut down on costs, we decided to multiplex. So basically, you'd have your sample, and each sample is tagged with an antibody that has a unique barcode. And once you tag your cells with that unique antibody, you can then just pull your cells together and makes it easier for sequencing, and also makes it cheaper when you're sequencing. So able to sequence an entire set of cells from one participant at the 3 time points, just using one 10x plate. So that makes it cheaper for us. And then this is just the sequencing workflow. So once I got the data, I run it through R using Seurat. And these were the cell populations that came up. There were quite a number, but I decided just, so once we got this data on R, we were able again to split it down by the different time points. So those, so each uniquely coded antibody, you can pull it out and you can get your different time points for each participant. And you can even go further down to checking the specific samples within each time point. So that was what we did. And then I decided to focus on specific cells rather than look at the entire population of cells that were present. So I chose to look at a monocyte, so both CD16 and CD14 monocytes. So they're heavily involved in controlling parasitemia, and as well they can contribute to dendritic cell populations because they're able to differentiate. And then I also looked at dendritic cells, the different population of dendritic cells, and CD4 T cells as well. So those are the ones that are mainly involved in antibody generation. So I decided to have a look at the differentially expressed genes between those who are able to control the parasitemia, so the chronic population, versus the febrile population, and just have a look at the different pathways or signaling events that are happening within these cells. And for the CD14 monocytes, what we saw was in the febrile type 1 interferon response was seen at day 9 post-infection. And at day 14 post-infection, you don't see anything, any signaling within these cells, but what you do see is upregulation of tryptophan metabolism. So it's a gene called IDO1, and it's the first step in tryptophan metabolism, and its byproducts normally inhibit monocyte function. So that was what we saw at day C14. At 14 days post-infection, and then looking at the chronic population at day C+9 and at day 9 and day 14 post-infection, we had this consistent type 2 interferon response. So interferon gamma was heavily involved in this group from day 9, and you still see it at day 14 post-infection. Another interesting thing that we found was the different ligands that are being activated within these CD14 monocytes. So in the febrile, we had the toll-like receptors being activated, and very many signaling cascades. So what was interesting was within each signaling cascade, there was activation of a negative feedback loop. So these molecules here actually inhibit some of these downward signals just to ensure that inflammation is kept in check. And in the chronic population, we actually saw that alarmins, so S100A8 and 9 were the ones pushing signaling through TLR4. And they were also, and what was being activated in terms of the transcription factors were actually similar. It's just that the pathway is being activated, the signaling path is being activated was different. So that was quite interesting. And then again, looking at the CD16 monocytes, it was the same again. But this time in the febrile, at day at day 9 post-challenge, we saw actually both type 1 and type 2 interferon being upregulated, which was quite interesting. Considering that CD14 monocytes, we only see type 1 interferon being type 1 interferon signaling being activated. And then in the chronic population, you still see your type 2 interferon together with TLR stimulation. And what was interesting in the chronics, in this chronic population, is that this CD16 monocytes are also being pushed to differentiate into dendritic cells. So that was quite interesting to see. So it's just a summary of both figures. Then again, looking at CD4 T cells, so this is both population of CD4 T cells. They had quite a similar expression, genes being expressed. So in the febrile population at day 9 post-challenge, you don't see any activation signals in the CD4 T cells. These signals actually pick up at day 14 post-challenge, and they're not type 1 interferon, rather they're type 2 interferon. So in these febrile participants, you see a shift from type 1 interferon to type 2 interferon, but this is much later on during the course of the infection, and at this time point, the parasitemia is too high. So whether this affects the immunity, this doesn't really affect the immunity, because at that time point, the parasitemia is too high, and they're being taken off the CHMI study. While in the chronic, what you see is CD4 T cells are being activated by APCs. So your DCs are able to activate your CD4 T cells as through the TCR receptor ligation. And then you get this activation of various pathways. And it's all of these pathways that keep coming up. So the MAP kinase pathway and F-kappa beta pathway. And you get the upregulation of AP1, which is the dimer of Gen and Phos. But then, and similar to the other slides, what you see is every inflammatory signal is negatively regulated by different molecules, just to ensure that inflammation is kept in check. So that was what we saw in both CD4 T cell population. So in the febrile, we see the type 2 interferon gamma signaling at day 14. At day 9 post-challenge, we don't see any signals or any activated pathways. And for the chronics, DC T cells are able to interact and activate CD4 T cells. So this figure just summarizes what we see in the genes being activated in the two groups. So in the febrile, it's mostly type 1 interferon pushing the response, and this and type 1 interferon is basically immunosuppressive. So it causes your dendritic cell to produce IL-10, and IL-10 in turn inhibits CD4 T cell. It doesn't inhibit CD4 T cells, but rather it pushes CD4 T cells to Tregs, and these Tregs secrete IL-10, and IL-10 just dampens the entire immune response, and that's why you see this slow peak of parasitemia over time. And also type 1 interferon can also decrease the interferon gamma receptors on cells. That's why with the CD16 monocytes, you see both active type 1 and type 2, but the type 2 interferon gamma is a bit weak. So that might be one of the reasons for that. And in the chronic participants, type 2 interferon drives a lot of responses. So for one, it drives the CD14 monocytes to produce IL-1 beta, and IL-1 beta is actually pro-inflammatory cytokines that activates dendritic cell function. At the same time, we have secretion of IL-8 by the CD14 monocytes, which recruits neutrophils to near the site. And once these neutrophils become activated, they secrete the alarmins that were present in the TLR4 signaling pathway. And these alarmins are actually quite potent at signaling than normal malaria GPI. So it initiates a quick response, but then that response is quickly shut down once the signal disappears. And then again, we also see differentiation of CD16 monocytes to dendritic cells, and we get the activation of CD4 T cells to type 1 Th1 helper cells, and they secrete interferon gamma, and that's actually good for controlling the malaria parasites. So in summary, so type 1 interferon seen in the febrile participants is immunosuppressive, as it inhibits dendritic cell function and impedes CD4 T cell activation. So this again is the reason why you see late activation of CD4 T cells, while in the chronic participants, interferon gamma is seen throughout all the time points. What was interesting is the alarming signal. I think we need to look more into that, into how alarming signaling affects CD4 T cells, and basically the dendritic cells in these participants active at both days, at day 9 and day 14 post-challenge. With that, I think I'd like to thank my supervisors, James, Paul, and Dr. Thomas Otto, for all the help that they've done, as well as Glasgow Polyomics, who did most of the sequencing, and my supervisor at the Cameron Wellcome Trust, Dr. Francis Ndong.